if you just stop the touch recorder, it's very counterintuitive, but if you click on, yeah.
Anyone need to stretch, coffee or donut, please do. I will see you through to lunch.
Okay. Give a round of applause for our next presenter, Dr. Molly Bright.
Thank you very much for the invitation to speak. It's a pleasure to be here. This is something a little bit different. I am going to be slightly evangelical about it. I really think that we should be understanding vascular function as much as neural function. This is particularly relevant for people who do fMRI for perhaps obvious reasons, but I am going to even talk about MEG. So for those of you who don't care about fMRI, please don't nod off. There's something in it for you. So I joined the faculty at Northwestern about a year and a half ago, and I run the Applied Neurovascular Imaging Lab there. So anything cool that I'm doing is thanks to all of the very clever people that you see shown here. We're based in downtown Chicago, and that environment is really a hub for rehabilitation research. And that's been a kind of new avenue that I've been excited to go into. Rehabilitation is, I think, an ideal area to be using multimodal approaches. Every individual patient is different. We're trying to track things longitudinally. We need quantitative measures, and we want to see how all the different aspects of physiology might be interacting during that period. I don't want it like that now. Yeah, he swapped it to this. It's correct on WebEx. All right, you'll see a couple of notes in the bottom where I copied some text, but enjoy the inner workings of my mind. Okay, so what I'm going to do is, I mean, I don't need presenter view, actually. It's fine. You can just put up the same. You do view. Nope. Yep. Beautiful. Thank you. All right, so what I'm going to try to tell you about is how a lot of us are already measuring some vascular signals when we're collecting other types of data. And usually this is with the aim of denoising what we're actually interested in. But I'm going to encourage you to amplify these noise confounds. And then by doing so, it can help us better differentiate different sources of signal variance in what we collect. And it also allows us to kind of see some new interesting things about how the vascular and the neural domains might interact with each other. Throughout, I'm going to scatter in some literature that demonstrates how the multiparametric data we end up getting from these types of experiments could be very relevant to a host of different clinical pathologies. And at the end, I'll leave you with a couple things that you could actually take home and try when you get back to your lab. So I think we typically, when we scan, just because it comes with the scanner, we often put that little photoplethysmograph on our participant's finger and we record the cardiac pulsation. And you can see that there's a fairly large effect around the ventricles, but that pressure wave is going everywhere in the brain, and we're seeing blood volume changes as a result. And so typically we use something like retroichor maybe to model these effects and remove it from the data. Something a little bit less standard, but becoming a little bit more common, is to also try to measure arterial blood gas levels. This is because carbon dioxide is a very potent vasodilator. When your CO2 levels go up, your vessels dilate, and this directly influences the bold signal. I'm not suggesting you use an arterial line with all of your participants. We estimate blood gases by measuring the content of what you exhale. So with just a little plastic tube under your nose, a nasal cannula, we can get an estimate of your arterial blood gases. And here you see an end-tidal CO2 trace, and then a very similar set of patterns in a bold fMRI time series. And so again, we measure it, we can model it, and we can remove it from the data. And that's what most people are going to want to do. But in addition to that, I argue that one person's noise is another person's signal. If you're getting this information, almost all of this is going to tell you something interesting about the brain if you just look at it differently. So here's a particularly elegant approach from Li Rongyang. She's using an arterial spin labeling type acquisition to measure arterial blood volume. And she's saying, well, okay, when the heart beats, you get a change in blood volume with the cardiac cycle. That's an artifact for fMRI. But if you measure the arterial blood volume at systole and diastole, which she's showing here in the left and the center columns, the difference of that change in blood pressure across the cardiac cycle, if you normalize that to the person's actual blood pressure readings, that's a measure of vascular compliance. And she shows nicely how that vascular compliance measure decreases with age. So this is currently a single slice acquisition. It takes ages. I don't know where she's at with it yet, but I think it's very exciting. But it doesn't really relate to fMRI, so I'll just put this out there as, see, noise can be cool. This is another example of not ready for prime time. Blood pressure fluctuations are always happening. We put someone in the scanner and they eventually kind of chill out. They're lying down in a dark space. And then you do see Meyer waves, so low frequency fluctuations in pressure. When I was in Cardiff, we built a device to modulate your blood pressure called a lower body negative pressure chamber, or a leg coffin, as we called it. We used a vacuum to decrease the pressure in the box. Blood pools preferentially in your legs, causing the pressure change. It's a tough experiment. You're getting sucked into a box. Head motion artifacts are a bit difficult to deal with. But Joe Whitaker has really been working hard to get interesting data out of all of this noise. So he's shown some interesting blood volume change effects having to do with autoregulation. And at the moment, he has some preliminary results looking at beat-to-beat fluctuations in blood pressure and how that manifests in our data. But again, you're probably not going to incorporate this tomorrow. Here's the thing I really want to focus on today. Carbon dioxide levels. They cause vasodilation, and it looks very similar to the bold effect we see based on neural activity. So we can use CO2 to derive this vasodilatory effect and use an fMRI acquisition to characterize that response. And that gives us this parameter called cerebrovascular reactivity, or CVR. Some people use the term vascular reserve. That's kind of the ceiling of how dilated could you get. Reactivity is just that change in bold or change in flow per change in CO2. And it's actually an umbrella term. It's the responsive vessels to any dilatory stimulus, so you could use acetazolamide like Diamox. But I prefer to use something that I can really play with in real time. So as I showed you before, just natural variations in your ventilation, your breathing patterns are going to change CO2 levels. So I could just try to capture that variance with a nasal cannula, and that's my CO2 stimulus. And certainly if you're just doing standard fMRI, this helps you denoise your data and might give you some insight into vascular reactivity. You could also ask your participant to amplify that variation, asking them to hold their breath briefly or take some deep breaths, and that gives you a slightly larger dynamic range. And then finally, if you want to be all fancy and complicated, you can have a very complex system. This here is the only commercially available one, but you could also build your own. But you can have a breath-by-breath computerized controlled gas blending system with a feed-forward physiologic model. You can give boxcars and ramps and sinusoids, and you can really play with CO2 the way you play with a lot of neural stimuli that we have. And that's me demonstrating just how much fun it is. So, I mean, ultimately, I think the theme of a lot of what I do in my lab is that, you know, however you want to use the information, we're going to benefit by being able to better understand different sources of variance in whatever data we collect. So however you want to look at my talk, hopefully it helps you in some way or another. But when we do things like cause vasodilation with CO2, it opens up a new type of experiment, and enter the calibrated fMRI experiment. So this is now how I'm going to link these vascular stimuli to measures that you're probably a bit more interested in about neural activity. So I likely don't have to motivate or explain why bold fMRI is a little bit insufficient all by itself to really quantify neural activity. But it's because of this indirect relationship, right? So we have a stimulus of some kind or a modulation of background activity, and there's neuronal activity associated with that we'd like to characterize. But that activity is actually coupled to a local hemodynamic response, which is what we actually measure with our acquisition. And this has some problems. So our typical fMRI experiment, we can map sort of where activation was happening and set thresholds for significance, but it's in fairly arbitrary units. And we have to assume a lot of these relationships in the middle, this neurovascular coupling and the hemodynamic response function. There are a lot of things that we can do in analysis to cope with variations in the shape of that hemodynamic response function, but the amplitude is a different thing. How can you tell what's a change in the amplitude of that transfer function and what's a change in the amplitude of the neural activity driving that response? So that's where we need to bring in a vascular stimulus. So I'm just going to describe the calibrated fMRI with the same framework in a slightly different set of schematic blocks. So we have a stimulus. It has some brain activity associated with it, and that's what we're interested in, but we can't actually measure it. But we know that brain activity is coupled to a blood volume change with vasodilation, which is linked to a blood flow change. And then there's also a change in oxygen metabolism, sort of rebalancing all of the ions and stuff. So all of those three things are happening, and they all combine with these baseline parameters here. So aspects of your sequence acquisition, baseline blood volume, hematocrit, all sorts of different physiological parameters. And they all go into this black box, which we can kind of model fairly well, and it gives us the bold signal we're actually recording. But it's a mixture of all of these processes. So we know we can measure bold. That's fine. We also know that we can be pretty reasonably good at simultaneously measuring blood flow with a combined ASL and bold acquisition. So that's covered as well. Then we kind of assume a relationship between blood flow and blood volume. So, okay, we can make that work. We still have two unknowns. We can't measure that metabolic change, which is our best estimate of the neural activity. It's kind of the most directly related to the activity because we have all of these baseline parameters influencing our signals. So what we need is something that drives the system but doesn't change metabolism, and that will let us fit for all these baseline parameters. So we need an isometabolic stimulus. And within reason, and I'm going to show you that I'm lying to you a bit later, but within reason, carbon dioxide is isometabolic. So we can drive a vascular response and kind of fix that CMRO2 variable. It lets us fit for all of these unknown parameters, and that's what I mean by calibration, because then we can use those parameters to then, in a second experiment, measure the metabolic impact of a task. So we're using bold and CBF responses to a vascular stimulus to calibrate the bold signal and allow us to estimate CMRO2, the cerebral metabolic rate of oxygen consumption. So there are two flavors of it. Calibrated fMRI, we give you a CO2 challenge, and that only lets us get relative changes in metabolism during a task. So we need a task that activates the region we're interested in, and in that region we could get a relative increase in metabolism. Sort of an up-and-coming approach is a dual calibrated fMRI experiment where we give two gases. We give carbon dioxide, we also give oxygen, which modulates parameters in a slightly different way. And that will allow us to estimate actually baseline metabolism. This is the relationship. You'll hear people talk about the Davis bottle and whatnot. You don't need to know the details at this point. I'm just going to talk you through it conceptually. So we see our bold signal is some relationship between volume, flow, and metabolism. That M is our calibration factor, reflecting all of that baseline stuff we don't really understand or know a priori. That beta value reflects aspects of how changes in deoxyhemoglobin actually influence relaxation times, and we get that from simulations, and people have their preferred simulated value. So there's a little bit of discrepancy in the literature there. And then we use what we call the Grubb constant as a relationship between flow and volume to further simplify this relationship. So again, we start off not knowing M or anything about metabolism. During hypercapnia, we can assume that CMRO2 is not changing. We can measure bold and flow simultaneously, so we can get M. And then we go back, use that M, and can actually measure metabolism. So what do we get? This is what we get. We get multi-parametric information. This is a hard experiment to do. It's at least 10 or 12 minutes. It's incredibly noisy. You have to wear a mask, and not all patients want to comply with that. I think we're getting better at understanding how to fit these data and how to deal with uncertainty and interactions between different parameters in the fit. On the left, you see work that's mostly coming out of Oxford, and on the right, you see more recent work using a Bayesian approach coming out of Cardiff. So what parameters can we access? We can access blood flow, that calibration parameter, which is kind of telling you what is the maximum bold signal you could possibly get in a voxel. We can get baseline blood volume. We can get that vascular reactivity metric that I've been talking about. And we can get oxygen extraction fraction, which is the key. So that's how much oxygen is the tissue pulling out as the blood is passing through. If you combine that with blood flow, you have tissue metabolism. And then we can get some dynamic properties like arterial arrival time. I think in the most recent versions, Mike Gramuska is even pulling out capillary diffusivity, so oxygen diffusivity. So we're getting closer and closer to measures that you can kind of get with triple oxygen PET and things that you can do with more invasive techniques in animals. And we're starting to be able to really estimate them in the whole brain in humans in a fairly reasonable experiment, So when we have that type of toolbox, what does that give us? So we look a lot to the PET literature because they've been getting this multiparametric information for longer than we have in MR. So here is an 015 PET paper where they're looking at traumatic brain injury and trying to predict outcomes looking at all the different parameters they get. And they say, it's important to emphasize that none of the physiological variables alone can be used to define acutely the extent of irreversibly damaged tissue. Future attempts to produce predictive maps for tissue outcome may require using combinations of these physiologic variables. And I think that's the ethos for this section. It's that one variable alone does not pin down particularly useful information, but putting them together, I think where we're really going to see advances is in being able to identify where an individual patient is on a scale of progression, because these parameters change in different ways at different times in different diseases. And so maybe we can really identify where this one individual person is on that trajectory. So an example, I think stroke is a really obvious application for this. We know that there are changes in blood volume through autoregulatory mechanisms to try to maintain flow and oxygen supply to the tissue, but eventually that's insufficient and you start to see real damage to the tissue. And so Derdine was just demonstrating that you can use CBV and OEF maps, and relative relations of that helps you figure out where you are in that curve. How close are you to that point where your autoregulatory abilities are going to be at their limit and you really are going to start to see some tissue consequences. In acute stroke, this is in animal models, you see that CBF was enhanced laterally for about a week after the injury, but that resolved in 21 days. But then reactivity, the ability of the vessels to actually respond to a new stressor, a new stimulus, that remained hyperactive through that 21-day period. And they argue that combining blood flow and vascular reactivity identifies a window where maybe specific types of therapeutic interventions might be efficacious. Alzheimer's disease, I think we're really building up an interesting body of evidence for how different vascular parameters progress alongside different symptom progression. So very early on, I think this is a really great study out of Oxford. Sonasuri showed that in very young, basically grad students, healthy young grad students who just happened to be ApoE4 carriers had already demonstrated impaired vascular reactivity in their early 20s. So we're seeing changes in vascular function well before there's any symptom or obvious consequence of those things. And I like the way one of my colleagues described this, which is perhaps an aspect of Alzheimer's or dementia is death by 1,000 cuts. You're always just a little bit shy of having the vascular regulation that you need to maintain healthy neural function. And then this other study showed that CBF was impaired very early in that blood flow or perfusion is impaired early in mild cognitive impairment, and blood volume changes were seen in much later stages. And so, this kind of puts together a picture of what happens when, and overall a feeling that treatments that could stabilize this or increase brain perfusion and reactivity could potentially delay some of the symptoms that we know are corresponding to this progression timeline. One study looked at one of these types of interventions from a vascular perspective. They showed that giving sildenafil, which I guess is a candidate therapy in Alzheimer's, it did increase global CSF, and then focally you saw, using a box-wise analysis, increases in perfusion in these specific bilateral medial temporal lobes, and that's done with an arterial spin labeling approach. However, they showed that vascular reactivity actually decreased, so I don't know how that connects to what I showed you previously. Somewhat, a bit of food for thought. They measured a global measure of metabolism. They didn't map it the way we can do with the experiments I was describing, but this global change in metabolism with sildenafil seemed to be related to their cognitive impairment as indexed by the MoCA score. So I'm not sure exactly what this means, but I think it's an avenue that more people are pursuing, because I think we have reached enough evidence that the vascular story that parallels or supports aspects of neurodegeneration and dementias and so forth, I think that story is becoming quite compelling. Multiple sclerosis, again, another disease that's typically described by hallmark issues in white matter tissue structure. You see these lesions being formed, but that lesion formation is actually preceded by a vascular event. So you see elevated blood volume and blood flow that predict the formation of lesions. That inflammatory effect does subside, and some lesions do resolve in MS, but lesions that go on to become chronic then reverse and show hypoperfusion, so reduced blood volume and reduced blood flow. We know when we're comparing MS with controls that vascular reactivity is impaired, so not only is there some change along with relapse and remission that is associated with these vascular parameters, but chronically we see vascular changes, and that might lead to some of the widespread global aspects of neurodegeneration that we also do see in MS. And then metabolism, as measured with a calibrated fMRI experiment, I'm happy to say, was shown to correlate with fatigue levels, and fatigue is this difficult to pin down but very key, common, important symptom in multiple sclerosis. So again, another pathology where adding the vascular element might tell you something more about what you're seeing in terms of neural activity. So I told you we can give vascular stimuli just like we can give neural stimuli. I've given you ways that you can try to separate these effects in a bold fMRI experiment to really look at metabolism versus reactivity versus flow and volume. And now I'm going to tell you you can't. So ultimately, thinking in silos never really does anybody much good. These neural processes and vascular processes are always going to be interacting. So I spend so much time trying to isolate and differentiate and put things in their baskets. And actually, there's some really cool phenomenon that happens because they interact all of the time. And so you could say the fact that bold fMRI measures vascular and neural properties is a curse, but maybe it's a blessing if you're interested in looking at that relationship. So, silver lining. So I promised MEG. I've been a part of a couple MEG experiments. It was eye-opening. So we gave graded hypercapnia challenges during MEG acquisition. So you can see here, that's the end-tidal CO2 data, and you see it going up from +2. So that's a +8 millimeter of mercury challenge, a +2, a +6, and a +4, with periods of just resting breathing in between. And we measured things like breathing rate and depth. We paid attention, they scored, how they felt about the gases. So we have a bunch of other metrics. We were trying to really pin down what do we see in MEG to do with the carbon dioxide specifically. And what Ian Driver and Cardiff showed was that we see a decrease in power in alpha, beta, and gamma. It's kind of widespread, and it seems to be sort of titrated with what CO2 level we're giving them. So we are changing your beautiful measurement of neural activity with our vascular stimulus. So I think the extra fun bit is that those periods of rest in between the gases that natural fluctuation in CO2 that happens because you breathe a little bit randomly at times, those natural fluctuations are sufficient to see corresponding changes in some of these oscillatory powers. So, you know, what you want to do with this, I'm not sure. Maybe everybody doing MEG and EEG should measure all of these vascular things too. You can denoise your data just like we have to with fMRI. And maybe you could also view it as there's something really interesting there to understand. So measuring it could give you, as we found with fMRI, it can maybe give you a new metric that's worth pursuing. I'm also interested in how the relationships of these neural and vascular aspects interact with, in terms of spatial patterns within the brain. Is it sort of what we saw in MEG, which was the whole brain kind of showed something together? If we look with fMRI, are we going to see that relationship show any more specificity than that in terms of spatial distribution? So this started with a little bit of a moment of kismet here. I just happened to be looking at some data involving breath holds. Breath holds increase CO2, you see a bold signal increase. And I was averaging together a lot of data and exploring what was in it using ICA. And this is work I did in Cardiff with these three. So, you know, the default mode network, it always pops up no matter what you do. But in my breath hold data with my decomposition, I found two networks that very closely resembled the default mode network. And when you look at the time series, something kind of interesting starts to appear. So in shaded red, you see where the breath holds of the data were. And the top network time series, you see a signal increase that kind of peaks a bit after the breath hold. And that's what we expect from a vascular reactivity effect. CO2 vasodilation, there's a little bit of a delay, and it will always peak after the breath hold is done. And we see these amplitudes are kind of reflecting the duration of the breath hold as well, which is kind of what we expect. And in red, I'm plotting what the end-tidal CO2 trace is, and you can see it fits very nicely. And then the bottom, we saw a slightly different type of time series, and it got me thinking. So what I kind of, if you squint, you can see it. You see perhaps a signal decrease during the breath holds, and that kind of made sense to me as well. It's a task we're asking them to manipulate their breathing. There's a countdown, they're paying attention. So deactivation of a default mode network seems sensible to me. But I have two of them. I have two of these networks, one showing me my vascular stimulus and one showing me an aspect of a neural stimulus, both present in my data. Why am I seeing a vascular stimulus specifically pulling out a neural structure? So to look at that a little bit more systematically, I came up with an experiment that had two neural stimuli, a working memory task a bit more formally than what we saw in the previous sort of data set, and a visual stimulus. And then I also gave these 4 blocks of carbon dioxide inhalation. And I collected this in 30 scans, and I did what I did before. I averaged them all together and had a look at what's inside. It was easy to find networks that look like my neural stimuli. You can see here the default mode network, task positive network, and visual network pop right out. That's kind of to be expected. But then what I saw was for each of these, there was a pair. There was a partner. There was another network that came out of this decomposition that wasn't identical, but looks fairly similar and has a fairly strong Dice coefficient. And when we look at the time series of these, it's not completely separate, but you do see a CO2-related effect. You see those 4 blocks of hypercapnia are the dominant effect in those time series. So I find this interesting to think about. And I think how you interpret it, even I haven't fully come to terms with what this means, because fMRI alone is probably not the right way to look at this. So if anybody wants to help me pin this down, please let me know after. But things we could think about are, do we actually see network-specific behavior of this cerebrovascular regulation, and what would that mean? So one possibility is that the vasculature is going to be coordinated with spatial patterns that mirror known neuronal networks. This could come about through development. We know these processes do track each other during development. So areas that have similar amounts of glial cells and similar endothelial function and whatnot might be matched across regions with similar neural function. So we see a relationship in space between vascular regulation and neural correlation. And it would provide perhaps the most efficient hemodynamic support for these neural networks if the vasculature could just be sort of constructed to supply specific networks with dilation. And then I think the other interpretation, which we can't separate out at this point, is that cerebrovascular function may modulate certain aspects of our neural networks. And this could be responsible for splitting those networks as, you know, maybe that's what I'm seeing. And as I think other papers, Braga and Buckner, are showing 2 default mode networks. I'm not saying one of theirs is more vascular than the other, but it's possible that vascular neural interactions are leading certain structures to be disassociated in our results. And if anybody wants a fun read, this is a great paper. It's called The Hemoneural Hypothesis. And it just demonstrates that the vasculature really can modulate excitability of neural circuits via chemical, physical, and thermal mechanisms. So basically, the vasculature passes the Turing test and can contribute to information processing in the brain. So whether or not we can really say which of these is happening in my data, I don't think we can at this point. But I think it's a very interesting area for further work, possibly bringing in additional modalities. And the other thing that came out when I was trying to learn about this area is this concept of the vascular neural network. This is being proposed as maybe more relevant than the neurovascular unit when we're thinking about pathology. So in this paper, they propose to take the neurovascular unit, which is this, you know, the center area here, but also include upstream arteries, downstream veins, perivascular nerves. And it's really all of that together when we look at the function holistically that gives us better insight into Parkinson's, Alzheimer's, et cetera. So this is another paper potentially worth a read. I'm particularly interested in how these relationships, and particularly these network-specific relationships, might develop. So we know from this great study with CO2 challenges that vascular reactivity is still increasing during adolescence, and it peaks around 15 or 16, and then I don't know, I'm not sure if it plateaus or begins to decrease at that point. Blood flow, on the other hand, is decreasing. Metabolism has already peaked much earlier. So what is that enhanced vascular regulation doing? Is it really a global effect, or is it supporting specific networks? So what I'm looking at now is tracking spatial distributions of vascular regulation and looking at how that corresponds to some of the changes in functional connectivity and structural connectivity that we see. I think this will be particularly interesting for understanding plasticity after an injury very early in development. So in my department, we work a lot with pediatric onset hemipresis. So we can see how an injury, perhaps prenatal versus shortly after birth, how that affects these different trajectories. And can we track changes in reactivity and flow and all of the functional aspects in our data. Yes. So I recently was exposed to some work by Andre Abenows, who's looking at changes in the vasculature in animals with traumatic brain injury being induced. So you see here, there's a sudden drop in vessel density at the site of the injury that then kind of resolves within 2 weeks. So the density goes back up, but the morphology of the vessels is not the same. So I think the vessel lengths are shorter, there's a shorter number of connections, all sorts of different parameters of complexity don't seem to actually be the same as in sham or pre-injury. And the blood flow to the region, despite the fact the blood volume is kind of back to normal, the blood flow is not back to normal. So structure does not equal function in the case of the vasculature. So again, we want multi-parametric aspects to really pin down what's happening after these injuries. So he, in particular, is comparing a lot of these vascular measures with tissue integrity measures, a lot of diffusion approaches. It's all in animals, and I'm now considering what I might try to incorporate into my own research. All right, last section. If I've bored any of you, wake up. This is the stuff you might actually be able to use. So if you're doing some MRI scanning, please measure CO2. It's a minimal cost investment, and it could potentially open up some really cool information. If you can, if you have the time and you have the participants that are compliant, try to vary CO2 a little bit more than just those natural fluctuations. It's worth it. You get much better data. And if you want to do more complicated stuff, come talk to me. I'm happy to explain all the pros and cons. One thing we're doing is trying to improve a resting state fMRI acquisition with a really pragmatic addition. And the motivation was that we know there's some temporal delay between things we measure like CO2 and the data we have. We have to figure out that optimal shift in order to model that variance and remove it. But the blue dots represent resting state data. I divided it into the first half and the second half. You don't get the same parameters in two halves of the same data. It's just too noisy. As soon as you add breath holds, you get the same parameters in both halves of your data. So adding a little bit of extra variance of CO2 helps you pin down that variance in your data. So these individuals in my lab are working on this now. We have our sacrosanct resting state scan, and we just tack on a little bit at the beginning. And we just use commands over the intercom. So we're adding maybe 2 minutes of scan time, the cost of a nasal cannula, which is $1 or $2, and a little bit of time explaining to the participant what you're going to need from them. If you don't get it, it's no big deal, but we often get it. So we haven't fit these results. That's just plotting them on a normalized scale, and you see the gray matter average bold time series, and you see that end-tidal CO2 regressor. So we ask them to do 2 sets of deep breaths, and you get these big vasoconstrictive bold signal decreases. And that's what we can use to really pin down CO2 variance in our data. So we're doing this in cerebral palsy. So here's one participant, very overt pathology, completely able to do the task, big, beautiful changes in CO2. And when we map reactivity, we see that reactivity is actually perfectly preserved in that tiny ribbon of gray matter closest to that very enlarged ventricle. This will help give us some insight into whether or not fMRI is appropriate in some patients. If we can see that vascular response, we know there's a vascular response that we can measure in our fMRI data. We trust it more. I'm also pairing up with collaborators in Spain, Cesar Caballero and Stefano Moya. They have a data set. It's a bit reminiscent of the Midnight Scan Club. They're scanning 10 subjects, 10 sessions over 10 weeks. They got a bunch of task fMRI, resting state fMRI, and I said, could you add a breath hold scan, please? And they did. They also got a bunch of physiologic information at each scan session that we can compare to. And, you know, preliminary results, there's a huge amount of data to unpack, but these are CVR maps. Each column is a different week of that participant. And you see that there's variation. So this week they have just lower CVR values kind of across gray matter. And so the ICC for how reliable this vascular reactivity measure is, it's not great. But I don't mind that because I expect physiology to vary over your 9 weeks. And that's the point of getting this data. It's to capture that variance. And when we look at the spatial reliability of the patterns of reactivity in the brain, those are more reliable. So we're seeing ICCs of about 0.7. Compare that to, in one of my papers, I showed a spatial ICC of around 1, but that was sessions on the same day, in the same actual scan session. So I think, you know, we can really pin down regional variations in vascular function with a simple technique like a breath hold with surprising success. And adding it to a longitudinal study, you can't go back and get this information. So if it's possible to collect it, it could really add some value to a larger study. So my take-home themes here. Measuring vascular signal, it helps you denoise your data, so you probably want to do it anyway. But if you actually amplify these vascular signals, it could help you with denoising, but also open up the possibility of very complementary metrics to give you a more multi-parametric view of brain physiology. Calibrated fMRI experiments capitalize on some of these vascular stimuli. They bring in ASL, and with some modeling, we can pull out metabolism, and that's sort of as close as we can get with fMRI to really measuring quantitative neural activity. With this multi-parametric approach, we're approaching kind of what we get with PET, but now we're in a modality where we could naturally pair it with diffusion measures and connectivity measures, things that we don't do with PET. Also, triple oxygen PET is not particularly accessible. MRI and a lot of these approaches that I talked about today are actually very accessible. So we're getting to the point where this methodology using MRI, neural and vascular stimuli together can really add insight in several different pathologies. Isolating neural and vascular phenomena, maybe that's not the ideal direction to be heading towards. It's helpful at times to think about differentiation, but maybe a lot of the most interesting stuff is actually where they interact and sort of combine. And there are several practical additions that you guys could mess around with tomorrow in your experiments to bring some of these vascular measures and explore for yourselves how that interacts with your patients. So with that, I'd like to thank everybody at all of these different sites where I work now and where I've worked in the past, and some funding, and thank all of you. Oh, yeah. Yeah. Yeah, so the question was about exactly how some of these vascular things are influencing the neural activity. Does it have to do with arousal? And my guess is yes, in part. I think there's also just direct effects of pH, and probably things that we don't understand or have hypotheses for. Does that change how I view denoising? Potentially. I mean, it's like when you have any collinear things in your model. Do you want to be conservative, or do you want to kind of push for what you're rooting for a priori? So if you have vascular arousal type things, if that's what you're interested in, then you want to do something with your vascular physiology to make it orthogonal. So we can do that. We can clamp CO2 levels and clamp O2 levels so that we can avoid some of the issues you're talking about. It's not trivial. You need some fancy equipment, but that might be one way to get around it.
I'll go to the microphone so people can hear online. I have been particularly fond over the years of doing EEG studies and fMRI studies at separate times on the same subject. And there's been something that's bothered me for years, which I've never publicly admitted, and I'm going to do it now.
We're honored.
Everybody.
We are honored.
And that is, we do fMRI studies lying down.
Yeah. Oh, yeah.
And your spatial ICC stuff demonstrated beautifully that pooling in the posterior brain. We do EEG studies sitting up.
Mm-hmm.
Okay, and then we compare the data. So can you comment about that?
Yeah, we thought a lot about this with our blood pressure manipulations, because lying down versus sitting up or standing up, that is one of the biggest daily blood pressure things that we do on a regular basis. And there are upright scanners, so we could give it a try. We were thinking about, well, if this lower body negative pressure thing doesn't work out, we'll scan people in a couple different scanners with a couple different positions. I don't know how well that would work. I do think lying down changes physiology. At least with lying down in an MRI scanner, it's a bit more similar than sitting up. I think in terms of consistency over sessions, if you're always lying down, it's always lying down. But what I would say is if you think some of those physiological changes that happen from the moment you lie down and then the next hour, that time course is going to maybe dictate the order of scans that you want to pursue. Maybe there's a way to measure when you think you've at least stabilized due to the changes that occur from lying supine. CO2. Done. So if you measure respiration, which is really ventilation, with a belt around the diaphragm, chest, then you are going to either directly measure non-vascular things or indirectly try to get a surrogate measure of those CO2 changes that you might as well just measure directly. So from the belts, you're getting, actual direct head movement, you're getting field shifts, but you're not measuring the vasodilatory effects of ventilation directly. So there are estimates with RVT and approaches like that, and now there's an attempt to look at flow changes rather than looking at chest position. And they work. I think I'm not entirely sure about how distinct that information is from what we could get with just a direct CO2 measure. So in my mind, it's not a huge expense, and I'd like the specificity of the CO2 measure. And in terms of the variance it induces in the brain, it also depends where you're looking. If you're looking at cortical gray matter, get a CO2 measure. If you're looking at the brainstem, maybe I'll change and say